Okay, so this is the pathway that we started with. Like I said, it has the content in there, but it is a little messy. So what we're going to do is start over. But before we do that, if I look at what I could change here, these are my enzymes. So I want to make sure it's clear that these are things that are modifying p53. I also have here that this is a transcription factor, so I want to have a way to indicate what transcription factors are and what they do. And then this is pretty clear where we have our outcomes. But let's start one more time. So we have DNA damage here at the top. We're going to have it activating p53. And we know that p53 can exist in two states. So remember how we drew enzymes over arrows when we did reactions? We do something similar with signal transduction pathways. So the arrow here indicates a change in state. And if I put AT over that, that indicates that the acetyltransferase is causing this change. And now I put my acetyl group on there. We also have, on the other hand, a deacetylase, and this one has no acetyl groups. Okay, so we can also do our little A in, A out here. The other thing we want to take care of is that these are transcription factors. So the way that I do this is I just put that protein right on the DNA, and the gene that it upregulates is BAX over here. And over here, the gene that it upregulates is p21. If you have BAX, we lead to apoptosis. And if we have p21, let's fix that here, we will have inhibition of the cell cycle. So this is really showing the same thing as before, but it's a little clearer. So this is a way that you might want to consider if you have anything that's in two states, is to draw something like this. You want to limit how many times that you draw any particular protein, but if it's doing something different when it's modified or not, then it is appropriate to show, for example here, acetylated and deacetylated p53. I hope that helps with your pathway drawing.